Imagine a massive, bustling factory. There are thousands of complex machines, conveyor belts running at super high speeds, and workers moving in absolute perfect harmony.
Right. It's a marvel of engineering.
Exactly. It really is. Now imagine the master control switch for this entire assembly line gets permanently jammed in the on-amp position.
Oh, man. The factory just goes into absolute overdrive.
It does. It starts producing relentlessly, consuming every resource around it, expanding its footprint until it eventually, destroys the very city it was built in.
It's a terrifying image to start us off, but it is an incredibly accurate description of what happens at the microscopic level inside a human cell when things go wrong. In this scenario, that factory is a cell in your body, and that jammed control switch is a specific protein called KRAS.
And KRAS is infamous, right? It's the notorious driver behind some of the absolute deadliest cancers we know of. But today, we are going on a deep dive into some source material that is completely shaking up how we understand this disease.
Yeah, it really is a paradigm shift. We're unpacking a groundbreaking 2026 paper from the journal Cell.
Right, and our mission for this deep dive is to explore a completely new mechanism for how this jammed switch actually operates. Because as it turns out, KRAS doesn't just act as a single broken switch floating around in the dark. It actually forms organized biological flash mobs inside the cell.
Which is such a wild concept. So to give you a roadmap of where we're heading today, we're going to dissect what these flash mobs actually are. We'll look at the specific molecular mechanism that causes them to form, how these organized gatherings physically supercharge cancer growth, and finally...
And this is the crazy part.
Right. The shocking discovery that a very common household cholesterol medication might actually be the key to breaking them apart.
It's just mind-blowing. So I want you to imagine looking inside your own cells right now. Forget the old textbook diagrams you might have seen in high school where the cell is just this watery soup with a few floating parts.
Yeah, those really don't do it justice.
No, not at all. Imagine it as a highly organized, dynamic, living city. To understand this new research, we need to start with the main culprit, KRAS. What exactly is this protein doing when the cell is actually healthy?
Well, KRAS belongs to a family of proteins we call small GT passes. The easiest way to think about it is as a molecular relay switch.
Sure.
Under normal, healthy conditions, it cycles back and forth between two very distinct states. When it's bound to a molecule called GDP, it is inactive. It's essentially sleeping, just quietly waiting for instructions.
Right.
But when it receives a growth signal from the outside of the cell, it drops that GDP molecule, grabs onto a different molecule called GTP, and wakes up. In this awake, active state, it broadcasts signals deep into the cell, telling it to grow and divide.
A good mnemonic I used to remember this is GDP is asleep, but GTP is awake and free.
Oh, I like that. It perfectly captures the cycle, but the massive problem arises when the KRAS gene becomes mutated.
The jam switch.
Exactly. The mutation essentially destroys the protein's internal timer. It loses the ability to turn itself off, so it gets stuck permanently holding that GTP molecule. It stays awake, constantly screaming at the cell to divide.
And the clinical burden of this specific jammed switch is just staggering. KRAS is mutated in 90% of pancreatic cancers, 50% of colon cancers, and 30% of lung adenocarcinomas.
It is a colossal issue in oncology, which brings us to the first major mystery unlocked in this Cell paper.
Right, let's get into the paper.
The researchers started by looking at human colon cancer tissues. They had 86 pairs of tissues, cancerous tissue, and matched healthy tissue from the same patients.
Okay, so a direct comparison.
Yeah. They used a technique to dye the KRAS proteins so they could visibly see them under a microscope. And what they saw completely challenged the basic assumptions we've held for decades.
Because logically, the assumption has always been that the more KRAS protein a tumor produces, the more aggressive the cancer would be, right?
Exactly. But when they analyzed the data, the total amount of KRAS didn't actually predict the tumor stage, the grade, or whether it had metastasized.
Wait, really? The sheer volume didn't matter?
The sheer volume wasn't the determining factor. What mattered was where the KRAS was located, and specifically how it was physically organized. In the most aggressive cancer tissues, the KRAS wasn't just floating evenly around the cell, it was clumped together, forming these dense, localized droplets.
Okay, which introduces a really fascinating physical concept called liquid phase separation, or LLPS.
Yes, LLPS.
The best analogy I have for this is making a vinaigrette in your kitchen. You pour olive oil into watery vinegar, And no matter how violently you shake that bottle, the oil eventually separates.
Right, it beads up.
Exactly. It forms these distinct spherical droplets suspended within the liquid. That is exactly what KRAS is doing in the watery fluid of the cell. The proteins are suddenly organizing themselves into a flash mob in a crowded plaza, separating from the rest of the cellular fluid.
And the clinical evidence tied to these specific liquid droplets is profound. The researchers ran the data through statistical models designed to isolate specific variables.
Meaning they wanted to see if this clumping mattered, regardless of a patient's age or other mutations or overall health, right?
Precisely. And they proved that higher levels of these KRAS droplets directly correlate with advanced tumor stages, lymph node metastasis, and significantly poorer overall patient survival. The physical organization of the protein is an independent risk factor for mortality.
Wait, hold on. Let me push back on this for a second. I'm picturing other diseases where proteins clump together, like the amyloid plaques we see in the brains of patients with Alzheimer's disease.
Sure.
But those clumps are essentially dead, solid, toxic aggregates that just choke the cell. Why is this clumping actively helping the cancer cells survive and grow?
That is the vital distinction here.
Yeah.
These KRAS droplets are absolutely not solid dead plaques. They are highly dynamic liquid droplets. The researchers proved this using a fascinating technique called fluorescence recovery after photobleaching.
FRP. Okay, that sounds like a sci-fi weapon. How does that work?
It is incredibly elegant. So they tagged the KRAS proteins with a green fluorescent dye, so the droplets are glowing brightly under the microscope. Then they take a powerful laser and zap just a tiny section of that glowing droplet, bleaching the color completely out of that specific spot.
Oh, wow. So you are left with a dark, dead spot right in the middle of this glowing microscopic orb.
Correct. Now, if the droplet was a solid dead plaque like in Alzheimer's, that dark spot would just stay there permanently because nothing is moving.
Right, it's frozen.
But that isn't what happened. Within seconds, new glowing KRAS proteins rushed into that dark spot from the surrounding fluid, filling it back in until the whole orb was glowing uniformly again.
That's wild. So it proved that these droplets are bustling, highly active hubs. Individual molecules are constantly zipping in and out of the mob.
Exactly. So it's not a cellular graveyard, It is an active, buzzing command center.
Okay, let me just do a quick recap for everyone listening to make sure I had this whole picture straight in my head. We have KRAS, which is a jammed growth switch driving deadly cancers. But the real danger isn't just the sheer amount of KRAS present. The danger comes when these proteins undergo phase separation, like oil and water, forming these highly active, dynamic liquid command centers that actually drive the cancer's aggression.
You have it perfectly. It completely changes how we view the protein. It is a great reminder that the physical architecture and the organization of a system can be just as dangerous as the components themselves.
Right. And for you listening, think about the systems in your own life. Have you ever noticed how the location and organization of a resource can completely change its power, rather than just the raw amount of it? It's the same thing in our cells.
Exactly.
So this raises the obvious next question. Water and proteins don't just magically separate on their own. Why do these KRAS proteins suddenly decide to huddle together into these droplets in the first place? I mean, they don't have brains, they're just molecules.
To understand the why, we have to look very closely at the anatomy of the KRAS protein itself. At the very tail end of the protein, there is a section called the hypervariable region.
DHVR.
Right. And right at the tip of this region is a specific amino acid. It's a cysteine at position 185. What the researchers discovered is that the cell attaches a specific molecule called a farnesyl group right onto this cysteine.
I was reading about this farnesyl group, and the way I picture it is the cell handing the KRAS protein a very thick, greasy, waterproof jacket.
I love that analogy.
Because the inside of the cell, the cytoplasm, is mostly water. And this farnesyl jacket is a hydrophobic lipid modification. Hydrophobic literally meaning it fears water.
That is the exact mechanism. It comes down to basic physics and thermodynamics. Because these farnesyl jackets are so deeply water repelling and they are floating in a watery environment, they naturally huddle together.
Just to get away from the water.
Yes. It isn't even that the jackets are necessarily attracted to each other. It is that they are all mutually pushed together by their shared exclusion from the water. This provides the physical driving force that pulls thousands of KRAS proteins together into a single phase-separated droplet.
So if this greasy jacket is what forces them to huddle together, what happens if we take the jacket away? Did the researchers try, like, stopping the cell from handing them out?
They did, and the results were definitive. They went in and genetically altered that specific spot on the protein. They mutated the cysteine at position 185 to a different amino acid called serine.
The C185S mutation.
Exactly. And suddenly the cell's machinery couldn't attach the greasy jacket anymore. Without that farnesyl group, the flash mob completely dissolved.
Wow.
The KRAS proteins just floated around the cell randomly in the fluid. The phase separation stopped entirely.
But how do we know it was specifically the water-fearing nature of the jacket and not some other complex chemical reaction happening at that exact spot?
To prove it was purely about the physical hydrophobicity, the researchers did a brilliant swap experiment. They took away the farnesyl group entirely, but they replaced it with completely different lipid modifications from other proteins.
Oh, interesting. Like what?
They used a myristoylation motif and another one called a germinylation motif.
So basically, they just swapped the brand of the waterproof jacket. They gave it a different coat that also hates water.
Yes. And as long as the protein had some kind of extreme water-repelling modification on its tail, the flash mobs formed again. They even ran the proteins through computational software to visually map the surface of the proteins. It confirmed that the extreme water-repelling nature of these modifications is the sole requirement to force the proteins to cluster together.
It is beautifully simple when you break it down. I mean, it isn't some complex lock-and-key enzyme mechanism driving this structure. It is literally just the physics of water and oil forcing the biological organization of a cancer cell.
It really is. Basic physics driving pathology.
Let's do a quick 3-bullet recap here for everyone following along. 1, KRAS mutations drive cancer. 2, these proteins undergo phase separation to form active liquid droplets. And 3, this clumping is driven by a hydrophobic modification called farnesylation.
Perfect summary. Which brings us to the functional question. We have built the factory, and we know exactly how the workers are being physically forced to gather on the factory floor. But what is the actual purpose of this gathering?
Right, what nefarious plotting is happening inside this biological flash mob?
Exactly.
Because KRAs can't just cause cancer while floating in the middle of the cell's fluid. We established it as a relay switch. To actually broadcast its growth signals, which it does through these pathways with alphabet soup names like MPK and PRK, KRAS has to physically anchor itself to the plasma membrane, the outer wall of the cell.
Right.
If it doesn't reach the outer wall, it can't transmit the signal.
Precisely. And to make that journey from the inside of the cell to the outer wall, KRAS needs to be processed. It requires final alterations before it is ready for the membrane.
Kind of like putting the finishing touches on it.
Yeah, think of it as a final quality control check at a manufacturing organelle inside the cell called the endoplasmic reticulum, or ER. The key player at the ER is an enzyme called RCE1. RCE1's job is to snip off the very end of the KRAS tail, perfectly tailoring it for its final journey to the outer wall.
So the researchers wanted to see what was happening with this tailor. They stimulated the cells with a growth factor, basically telling them to grow. They used mass spectrometry to analyze exactly what proteins were hiding inside these liquid KRAS droplets, and they found the RCE1 tailor inside the droplet.
This is a phenomenon known as co-condensation. When the growth signal arrives, the entire KRAS liquid droplet actively moves to the ER membrane and physically pulls the RCE1 enzymes inside the droplet with them.
It is literally like the flash mob abducting the tailor. They grab RCE1 and pull it into their greasy huddle so they can mass produce the final uniforms they need to infiltrate the cell membrane. It creates a hyper-efficient processing factory right there in the liquid drop.
It's terrifyingly efficient.
But let me ask a practical question here. If we artificially melt the flash mob, say using that genetic mutation we talked about where they don't get the jacket, can KRAS still find the tailor and get to the membrane the old-fashioned slow way? Does the cancer absolutely need the huddle to survive?
That is the ultimate clinical question. Is this just an interesting physical quirk, or is it a vulnerability? The researchers tested this in vivo using mouse models. They took mice and replaced their natural KRAS with the mutated version that couldn't put on the jacket and couldn't form condensates.
And what happened?
The result was striking. The KRAS signaling, which they measure by looking at those PRAK pathways, dropped dramatically. The proteins simply couldn't get processed efficiently enough by floating around randomly. In these mice, the tumor growth effectively stopped.
Wow. So if the proteins can't build the factory floor, the whole assembly line grinds to a halt. The cancer cell is relying on the extreme efficiency of that droplet to process enough KRAS to keep growing. So a quick summary for you guys listening. The droplets act as we finally recently developed targeted drugs like sotorasib, also known as AMG 510, for specific mutations. But the clinical nightmare is that the cancer adapts. It becomes resistant to these drugs incredibly fast, sometimes in just a matter of months.
Yeah, the rapid onset of drug resistance is one of the harshest realities in oncology. The cancer almost always finds a workaround. But this paper presents a totally new angle of attack. Instead of trying to pry the switch off, the researchers asked, can we dissolve the factory floor entirely?
Okay, so how did they try to do that?
To find out, they took a library of 747 small molecule drugs that are already FDA approved for various other diseases. They tested every single one of them on human cells in a lab to see if any could melt the KRAS condensates.
I love these massive drug screens because the answers are always so surprising. The winners weren't some exotic billion-dollar new chemotherapies. They were statins.
Yes.
The exact same statins that millions of people take every single day for high cholesterol. Specifically, drugs like pitavastatin, simvastatin, torvastatin.
It is a remarkable finding. But to appreciate why it matters, we need to carefully explain the mechanism of why a cholesterol drug melts a cancer droplet, because it beautifully connects back to the biology we just unpacked.
Right, it's not random.
No, not at all. Statins work by inhibiting a specific enzyme in the body called HMGCR. This enzyme sits on a biological highway called the mevalonate pathway. Now, the main destination of this highway is the production of cholesterol. That is why patients take statins.
Right, you block the highway, you lower the cholesterol.
But there is an exit ramp off that exact same highway. A side branch of the mevalonate pathway produces a completely different molecule called GGPP.
And I'm guessing GGPP is connected to our waterproof jackets.
It is the crucial link. GGTP is the raw material the cell uses to synthesize those farnesyl groups.
Oh, that is incredible. So by taking a statin, you aren't just lowering cholesterol, you were literally cutting off the supply chain for the raw materials needed to make the greasy jacket.
Exactly.
If the cell can't make the lipid modification, KRAS doesn't get its waterproof coat. If it doesn't have the coat, it doesn't fear water. And if it doesn't fear water, the condensates dissolve back into the fluid. No factory floor, no efficient processing, no tumor growth.
It is a perfect cascade. And they proved it with a rescue experiment. They gave the cancerous cells pitavastatin, and just as expected, the droplets melted. Then they artificially added that raw material, GGPP, back into the cell culture. And right before their eyes, the flash mobs were formed because they manually restarted the supply chain.
Okay, this is fascinating biology. But we need to talk about what this means for overcoming that massive problem of drug resistance we mentioned earlier. How does melting the mom help a patient whose cancer has stopped responding to traditional KRAS drugs?
The researchers used a PDX model, a patient-derived xenograft. Essentially, they take actual human colon cancer tissue that has a specific KRAS mutation, and they implant it into mice. This provides a highly accurate model of human cancer. They treated these mice with the new targeted drug, AMG 510. At first, the drug worked and the tumor shrank. But then, perfectly mirroring what happens in human patients, the cancer became resistant. The tumors started aggressively growing again, completely ignoring the drug.
The cancer found its workaround. The individual machines kept running.
But then, the researchers changed the strategy. They combined the targeted drug, AMG 510, with pitavastatin. The statin traveled down the mevalonate pathway, cut off the raw materials for the farnesyl jackets, and dissolved the KRAS condensates.
And what happened to the resistance?
Without that highly efficient factory floor organizing the proteins, the cancer's resistance completely collapsed. The statin restored the cancer's vulnerability to the primary drug, and the tumors successfully shrank again.
Okay, I want to be very careful here and offer a clear caveat to anyone listening. Statins alone are not a magical cancer cure right now in human clinical trials. The data over the years on using statins as a solo cancer treatment has been mixed.
That's a very important point.
Right. But what this paper reveals is a massive, previously hidden structural mechanism. It points heavily toward a future of combination therapies. The idea isn't using a statin to kill a cancer by itself, but using it to dismantle the physical architecture of the cancer cell, so the actual targeted cancer drugs can get in there and do their job.
It represents a fundamental paradigm shift in pharmacology. We are learning that we don't just have to attack the active site of a broken protein directly. We can attack the social network of the protein. We can attack its ability to undergo phase separation.
So just to make sure we've captured the full picture of this breakthrough for our audience, by screening hundreds of common drugs, scientists realize statins can melt these dangerous cancer hubs. They do this by blocking the cellular highway that provides the raw materials for those greasy jackets. And most importantly, when tested in animal models, combining a common statin with a targeted cancer drug completely overcame severe drug resistance.
It leaves us with a truly profound realization. How incredible is it that a daily medication, taken by millions of people simply to protect their heart, might actually hold the master key to breaking down the physical architecture of one of the deadliest cancers on Earth?
It is mind-blowing. We used to think KRAS was just a single, isolated, broken switch floating in the dark. Now we know it is a dynamic, highly coordinated mob. And more importantly, we finally found a way to disperse the mob.
It proves that by understanding the absolute deepest details, the exact amino acid, the specific lipid jacket, the precise enzyme tailoring the protein, we uncover entirely new systemic vulnerabilities that we never would have seen otherwise.
Which leaves me with one final provocative thought for you to mull over as we wrap up this deep dive. If a medication as ubiquitous as a simple cholesterol pill can literally change the physical state of a cancer protein, melting it from an organized liquid hub back into a dispersed, harmless soup, what other common, safe drugs sitting in our medicine cabinets right now are secretly hiding entirely unknown structural mechanisms in our cells?
Oh, that's a brilliant question.
Right. What other phase-separated factories are we accidentally turning on or off every single day without even realizing it?
The molecular world inside us is infinitely more dynamic and interconnected than we ever realize.
Absolutely. Keep questioning the structures around you. Because as we saw today, understanding the deep details really is the ultimate shortcut to being well-informed. Until next time, keep exploring the muddy waters of the microscopic world.